بسم الله الرحمن الرحيم. You will study a very interesting chapter in biology. You will study enzymes together. Let's start our lesson. Before we start our lesson today about enzymes, we have to ask a few questions. What is the catalyst? We know that the catalyst is a substance designed to increase the rate of the reaction. Okay, is there any catalyst in our body or any living cell? Yes, there is a biological catalyst in the living cells and our body. Third question, why we need a catalyst in our bodies or other living cells like microorganisms? We need a catalyst to increase the rate of the reaction and to lowering the activation energy needed in the reaction. Fourth question, enzymes are a catalyst? Yes, enzymes are a catalyst which act to convert the substrate into a product by lowering the activation energy and increasing the rate of the reaction. Catalyst definition is a substance that increases the rate of a chemical reaction and is not consumed يعني لا يستهلك and not changes يعني لا يتغير by the reaction. يعني the catalyst enter the reaction to accelerate it and lower the activation energy without any changes for the catalyst. At the left here we see the reactant converted into products but consumes a big activation or a high activation energy. In the right here we have a lower activation energy consumed when we use a catalyst. Living cells have two types of proteins, functional proteins and structural proteins. Enzymes are proteins, but it is a functional protein. It helps to speed up our metabolism in the living cells, converting the substrate molecules into the product. Sometimes they build up some substances and others breaks down the big molecules into smaller molecules. All living cells have enzymes, but our bodies naturally produce enzymes as our bodies naturally produce proteins. How could this happen? How the enzyme work in our cells? Sometimes the enzyme present in the cell as inactive form or inactive enzyme called apoenzyme. Apoenzyme must be converted into holoenzyme, which is the active form of the enzyme. The cell converts the apoenzyme into holoenzyme by cofactors by three ways. Cofactor may be metal ion or coenzyme or metal ion and coenzyme. Metal ion and coenzyme are cofactors which convert the apoenzyme into holoenzyme to be in active form to enter the biochemical reaction. Enzymes are highly specific. This specificity comes from a specific site on enzyme called the active site. An active site binds with a specific substance forming enzyme substrate complex. This means the active site is the region of an enzyme by which or where substrate molecules bind and undergo a biochemical reaction. This means there is a high specificity between the substrate and the enzyme, and the active site is the region responsible for this specificity. There are two theories explain the action of an enzyme. First one is lock and decay model. In this one, active site must match the specific substrate structurally and chemically. Active site and substrate must fit each other and complement each other to form enzyme substrate complex. Without combination structurally and chemically, there is no binding between the substrate and the active site. This picture explains the K, which is the enzyme, and the LOX, which is the substrate, when binding together, complement each other to form enzyme substrate complex in order to form the product. The K, which is the enzyme, is get out the reaction without any changes to it.